So why are people interested in controlling chamber CO2 concentrations at very low CO2? Principally, it revolves around being interested in C4 plants and getting good estimates of the CO2 response in these plants. This provides significant insight into the PEP carboxylation and other biochemistry that is happening in C4 plants. Just as a reminder about how C3 plants differ from C4 plants, the carboxylation event in C3 plants is on a 3-carbon sugar, whereas the carboxylation event on C4 plants is on a 4-carbon compound. The initial carboxylation event is mediated by different enzymes in these two plants, the C3s and the C4s. The kinetics for these enzymes are very different, leading to quite different responses to CO2. The initial slope of the C3 and C4 responses give a measure of the rate of carboxylation. The CO2 compensation point for C4 plants is typically about 10 micromoles per mole, whereas in C3 plants it is usually 30 micromoles per mole or greater. As the global climate changes, increases in CO2 concentration and temperature shifts will alter the relative fitness of C3 and C4 plants. This will likely alter their distribution and make domestic cultivation more difficult. In the LI-6400, chamber CO2 concentrations can be controlled down to 30 to 50 micromoles per mole depending on machine. This makes it harder to assess the CO2 compensation point in C4 plants. By modifying your system, we are able to achieve a concentration of 1 to 2 micromoles per mole CO2 while still maintaining optimal conditions for gas exchange. In the LI-6400 system, CO2 is regulated by a voltage. The control voltage for the standard flow path and the modified flow paths are shown here, and they're slightly different. You can see about a 10% reduction in maximum CO2 achieved in the modified flow path, but what you actually give up at the top end, you gain at the bottom end, as shown by the inset. The modified flow path does have an achieved CO2 concentration that is about 0 and is a fairly continuous response, whereas the standard flow path does have a limitation at 30 to 50 micromoles per mole depending on system. The CO2 performance is not impacted by the modification. There is a loss of about 10%, as I noted previously, in the maximum CO2 concentration, but it's still greater than 2,000 micromoles per mole for most machines. The system noise is unaffected by the modified flow path, but you do have slightly longer times to stability when changing your CO2 concentration. We'd like to know why you're interested in making measurements at very low CO2 concentration. We've got a little survey, and if you'd click the radio button that corresponds to the answer that reflects your interest. If you do select other, please use the question and answer panel to let us know why you're interested in making measurements at very low CO2 concentrations. As expected, many of you are interested in C4 plants or C3, C4 intermediates. Before we begin, there are a few tools and supplies that you're going to need to gather up before beginning the modification. Typically, the first time a person goes through this modification, it takes about 2.5 hours. If you're doing multiple machines, you will become much faster at it at subsequent machines. You will need a supply of bevel-lined tubing. You're going to cut that into 4 sections. It's also good to have a supply of silicone or Teflon grease. These are normally found in the spares kit of your 6400. It's also a good idea to have some replacement hose barbs. Although they're not needed, typically hose barbs are damaged and should be replaced if they are, and having a supply of those will prevent you having a long downtime with the system. The tools that you'll need are a pair of side cut pliers, a razor blade knife or similar to cut the tubing, a couple of screwdrivers, the Phillips head screwdrivers, and a 1/4 inch or adjustable wrench to remove any damaged hose barbs. Before we start, we need to cover a few techniques and a couple of warnings to make the modifications easier. When cutting the tubing, you want to make sure that you have a nice, smooth, square cut on the tubing. Deformed tubes like this or ragged cuts like this result in leaks around the tubes and the hose barb connections. The cut should also be perpendicular to the flow path, unlike what's demonstrated here. When you have an angled cut, as this demonstrates, that does not match up to the hose barb, potentially leading to diffusional leaks. When bending your tubes, care should be taken to ensure that you have nice, gentle bends. If you overbend the tubes, as demonstrated here, this will result in a crease, and that crease causes a diffusional leak through the wall of the tubing. A technique that we've developed here at LI-COR to remove the tubing from hose barbs is to use a pair of side cut pliers and grasp the tubing at an oblique angle to the barb. Squeeze the tubing, do not cut all the way through to the hose barb, and then gently pull that tubing away, stretching it on the hose barb. This allows you to move the tubing away from the hose barb without cutting all the way through. Once you get that tubing stretched away from the hose barb, then go ahead and cut all the way through the tubing. This will leave a small fringe around the hose barb that can easily be torn off. Do not attempt to use a blade or other similar device to cut the tubing from the hose barb. The barb will be scored and must be replaced to prevent leaks. If you do damage a barb, no worries. They're easily replaced, and with a 1/4 inch or adjustable wrench, you can remove the hose barb from the fitting. When replacing it, be sure that you have a black ring around that hose barb. Now, just a word of caution. The hose barb on the outlet of the CO2 mixer, as shown here in the circle, has a very fine tube running through it. Be very careful of this rod that is inside that hose barb. The rod is a very fine glass tube that can be sheared off if you do remove that hose barb at an angle. You want to remove that hose barb from the CO2 mixer straight to avoid any damage. This is a picture of an intact hose barb. If this tube is broken, your instrument will need to be returned to LI-COR for repairs. We're now ready to begin the modification. To modify the instrument, you're only going to be working with the console, so remove the chemical tubes, the cable bundle, the power supply, and the erga head and set those aside. Remove the 16 screws from the console and lift the console carriage out of the shell. Turn the carriage upside down, being careful not to lay it on anything that's going to damage the screen or the keyboard. We're going to be altering only a portion of the flow path from the blue Ballston filter to the second buffer volume. We will want to remove the tubing and discard tubes 1 through 4 as labeled in this diagram. Although I demonstrate removing the tubing from the buffer volume disconnected in the carriage, you will want to be sure not to remove any of the components from your instrument. Remove the tubing from the installed components. Tube 1 is removed by pressing in on the orange or red fitting on the quick connect, which will release the tube from the quick connect fitting. Tubes 3 and 4 are more easily removed from the system by removing the pump. This pump is held in place by a single screw, which once the screw is removed, the pump can be repositioned and the tubing can easily be removed from the pump. You do want to take care not to damage the plastic hose barbs. If you do damage those plastic hose barbs, you will have to replace the pump. You will want to remove tubes 2, 3, and 4 from the system, and your instrument should look like this at this point. You're going to have some tubes still in place, but you have removed the 4 tubes to alter the flow path. We're now ready to begin installing the modified plumbing. You're going to need 4 new tubes to replace the ones that you've just removed. The links are listed here. The first thing that you want to do is you want to slide the new tube number 1 under the cable clamp on the side of the CO2 mixer. This location will protect it from damage by the shell when you reinstall the shell on the carriage. Connect the end of the tube to the quick connect. Silicone or Teflon grease will make this connection easier. Push the tube in until the tube is firmly seated. Connect the other end of the tube to the pump. You want to connect tube 1 to the inlet side of the pump. The inlet side and the outlet side of the pump are marked by an arrow that indicates the direction of the flow path. Then connect tube 2 to the outlet of the pump. Reinstall the pump and then connect tube 2 to the lower barb on the first buffer volume. Connect the new tube number 3 to the inlet side of the CO2 mixer and feed the tube underneath the tubes coming out of the second buffer volume. This location will protect this tube from damage when you reinstall the shell on the carriage. Connect the new tube number 4 to the lower barb on the second buffer volume and then gently form it into a clockwise corkscrew. This will prevent the tube from being twisted in such a way that will damage the tube or create a crease that leads to diffusional leaks. Attach the new tube number 4 to the outlet side of the CO2 mixer as shown here, and then the final connection is to take the new tube number 3 and connect it to the upper barb on the first buffer volume. You do this by gently forming a bend in the tube to connect it to the barb. Be careful not to overbend the tube and damage that tube, restricting the flow. The flow path is now complete, and it is different from the standard flow path as the instrument comes from the factory. This allows for control of the CO2 concentration inside the chamber at very low concentrations. The new tubes can be removed from the system at a later point if you so choose, and the system returned back to the standard flow path by simply reversing the configuration and putting the system back in the flow diagram as shown in an earlier slide. Once you've modified your system, it is important that you test it for leaks. You can accomplish this by righting the carriage, but do not put it back in the shell. You want to attach the chemical tubes, cables, ergas, and battery to the instrument. Once you've connected the power to the instrument, do be careful. We don't want anyone getting hurt by touching any of the internal components. Power the instrument on, being careful not to touch the internal components, and then with the system in new measurements, you want to gently blow underneath the carriage. This will allow you to see any spikes in CO2 concentration that may occur. You accomplish this most easily by setting the CO2 scrub to full scrub and the desiccant to full bypass. This will give you a zero concentration in both the analyzers, and so any increase will indicate a leak within the system. Now that we have completed the hardware changes, it's time for a little bit of a break. We'd like to know how comfortable you are with making the modifications. Please fill out the survey and let us know what you think about doing both the hardware modifications and how comfortable you would be in editing the software. For those of you that have never edited the software code, no worries. We're going to walk you through it step by step, and by the end of it you should be able to make these modifications without any problems. The modification to the software is necessary to accurately generate the user calibration curve like was shown in that earlier slide. The edits are pretty straightforward, and we're going to show the system in a new XT configuration. Any software that you have on your 6400, the files are all going to be in the same location, and there's going to be slight differences in some of the menus, but the software modification is identical as far as the things that need to be done. The software edit is necessary for any systems operating 6.1 0.3 or older. Newer systems, as shown here, have slightly different menus than older systems do, but all the steps are the same. The first thing you want to do is from the main home menu, you want to press the F5 to access the utility menu. In the utility menu, you want to expand the file menu and highlight the access the filer. You want to press F5 to select that. Once you have selected that, press F1 for the directory. You are going to change the directory you are looking at. You want to highlight the sys directory and expand that by pressing F1. Highlight the sys open directory and press F5 to select that. In the sys open directory, locate the file open.mxr. This is the calibration program for the CO2 mixer. Press E for edit, and you have now opened that CO2 mixer calibration coding. You're going to press the N key on your instrument and then press the page up key 23 times. That's going to take you to the middle of this file where we need to edit the coding. I have shown the modifications to the coding that we need to do here. It's only a portion of the file, and we're going to change those things that are underlined in red. In the 6400, you want to modify the mixer signal origin and mixer ppm lines. You want to add 50 and 0, and also add 15 and 0 to the end of those lines. And now you've completed the software edits. You want to press Escape, then you want to press U for Update, then press Q to quit the editor. You should be taken back to the utility menu. You want to press Escape in the utility menu, and that will take you back to the main home menu. From the main home menu, press the F3 key, and that is going to take you into the calibration menu. We need to now implement those software code edits that we have just done. In the calibration menu, you want to select the CO2 mixer calibration and press F1 to expand that in newer systems. Select Calibrate and press F5 to select that. This is going to open an automated program to run the CO2 mixer exercise. It's going to warn you that you want to make sure that the CO2 mixer is on. You want to put a CO2 cartridge in that CO2 mixer, and then go ahead and press Y for yes. You need to wait a little bit for the CO2 concentration to stabilize. Typically that CO2 concentration should stabilize at 2000 ppm or greater. If you have a much lower CO2 concentration than that, you do want to check and make sure that there is an active cartridge in your CO2 injector source. Once you've achieved a maximum CO2 concentration, go ahead and press Y for yes to accept that maximum CO2 concentration. And this is going to begin the automated program where the system steps through a collection of CO2 concentrations at different voltages. You do want to note that the coding should read 10 different set points. If that's not showing on your screen, then the edits that you made are not implemented, and you do need to stop the CO2 calibration and go back and make sure those edits are in place. Another thing that may happen is you may not have implemented those, and just simply restarting the system will implement those changes that you have made. Once you've completed the CO2 steps, the system will show you the values. Go ahead and press Y to plot this. You want to look at that plot and ensure that it is a smooth, mostly linear response. If you see any jaggedness to that, you do want to rerun that CO2 mixer calibration. Once you have a CO2 mixer calibration that you're comfortable with, go ahead and press Y to save that calibration and implement it. You're now ready to make some measurements. So we have completed the CO2 modification, we have completed the calibration, and the CO2 control is going to operate as normal as you've grown accustomed to in the 6400. In new measurements in the menu level 2, F3, you're still going to have the same choices for controlling on CO2 reference, CO2 sample, or on a constant millivolt setting. You still have control by 1 micromole per mole increments, and the CO2 concentration typically is going to be greater than 2,000 micromoles per mole on most instruments. The flow control for the modified flow path is going to be slightly different. We do not recommend using the automatic flow controls, example, the H2S control, the relative humidity control, or the vapor pressure deficit control, because of a momentary instability that happens when you change the flow rate. So with the modified flow path, momentary CO2 fluctuations occur every time the flow rate changes. I have done a quick little experiment here. You're looking at some data where the instrument was changed from a flow rate of 500 micromoles to 700 micromoles per second, and that change in flow rate resulted in a spike in the CO2 concentration in both the reference and in the sample cell. This spike in CO2 concentration results in a momentary change in the photosynthetic rate, and the problem is that this leads to instability. So we recommend using a constant flow rate and then using the desiccant to control the water concentration by altering the scrub or bypass adjustment. As a reminder, we recommend using a relative humidity between 50 and 70% in the sample chamber. The automated flow control is not recommended because it results in a change in the flow, which will change the CO2 concentration, which will change the assimilation rate, which will change the stomatal aperture, which changes the stomatal conductance, resulting in transpiration rate changes, which change the water concentration in the sample chamber, which result in the flow rate once again adjusting to maintain the constant water concentration or relative humidity that you requested. So we recommend using the constant flow rate to avoid these problems. The result is that when you look at photosynthesis rates in either a constant flow rate or a continuously variable flow rate, you end up with two different responses. You can see that in the constant flow rate control, the CO2 response of this individual leaf is very smooth, very uniform, and we get a nice response to increasing CO2 concentration. But when we use the variable flow rate with the modified flow path, we have a lot of instability that sets up in the system due to the CO2 rate changes, and this results in a very uneven, jagged response to increasing CO2 concentration. Now, just a little word about diffusion and low CO2 concentrations. We've made a modification which allows you to get down to very, very low CO2 concentrations in the 6400. When you're working at low CO2 concentrations, you're setting up a large gradient between ambient CO2 concentration outside the chamber and the CO2 concentration inside the chamber. And this can result in a diffusional leak that causes a resulting error in your calculated rate of photosynthesis. I would recommend looking at these authors and also at the 6400 manual, which has a method of correcting for the diffusional responses. To show you what we're talking about, I made a measurement on 3 different corn leaves. I did CO2 responses of these 3 different corn leaves at saturating light. And you can see that there's the uncorrected rate of photosynthesis and a corrected rate of photosynthesis. And that correction is small, but it's actually significant. When you look at the absolute error that is resulting from diffusion here on the very low end, low rates of photosynthesis, you can see that that absolute error is small. But if you look at it as a percentage error resulting from the diffusion, you can see that we're looking at almost a 200% error at the very lowest rates of photosynthesis, which also correspond to the greatest difference between ambient and chamber CO2 concentrations. So to summarize, we have made a modification to the flow path inside the instrument, we've changed the calibration, and we're now able to control CO2 concentrations at very low rates. There is the caveat: you do want to be conscious of diffusional leaks, and with that we'd like to thank you for participating, and as always the LI-COR staff is here to assist you with any questions you may have about making the modification.
Thanks, Pat. We'll take a moment to answer just a few questions. As a reminder, if you've asked a question that hasn't been answered, our panel will stick around until all of your questions have been answered. All right, Pat, our first question is, how bad do the marks on the hose barb need to be before I must replace it?
Thanks, Ashley. basically any marks that you can see that are on the hose barb are going to cause a leak around the tube and hose barb interface. And the bigger the mark, the bigger the leak. But we recommend if you can see a mark at all on that hose barb, go ahead and replace it. They're fairly easy to replace, and they're inexpensive, and it's going to give you much better measurements. The problem with leaks are they're going to be twofold. They're going to change the CO2 injection rate, and it's also going to be a leak that can affect your zero and span settings if you do the user zeros and spans.
Thanks, Pat. The next question is, this is more than I want to do. Will LI-COR modify my instrument?
Well, the conversion is pretty straightforward. I understand being a little hesitant to do this. Yes, we will modify your instrument if you want to send it back. There is a charge associated with doing that. But if you do the modification yourself, there are times that you may want to go back and switch back to the old system. There are some benefits of doing so. You have the automated flow control, which will let you control on RH and water control, water in the chamber. And so if you do it yourself, you can get familiar with that. We will switch it back for you if you want to call us and contact us for a return authorization number. And any of the LI-COR staff will be willing to assist you with that.
Thanks, Pat. And thank you, everyone, for your questions. We look forward to seeing you at the next webinar in this series.